Start the next session, and we're beginning with Peter Dearden, Dr. Peter Dearden from University of Otago. He's actually a professor nowadays. He's worked on bee research for many years, but also insects before that, other insects before that, and in honeybees in particular in the last sort of 12 years. His interests lie in the biology, development and genetics of bees, and he is the research director of Genetics Otago and the director of Better Bees Research. So him and Gerda will be speaking today. I'd like you to welcome them to the stage. Thank you.
Hello. I got promoted, so I have to wear a tweed jacket now. That's how it works. It's good, isn't it? I also, I'm going to do a double act today. So I'm going to talk about some of the work that we've already done, and then my PhD student here, Gerda Peterson, is going to talk about some of our future work, which we want to, some of the work that she's doing and some of the work that we want to do to try and improve bee stocks. It's really bright up here, so if I look like I'm squinting, it's because you're shining these lights in my eyes. My name's Peter. We're going to talk about a bunch of things. I want to start by saying thank you all for inviting us to come and talk. It's good to see so many of you here. And let's get on with it. So one of the things we've been interested, if I can press the button, does that work? Yes. Okay, so one of the things we're really interested in is how we can use our understanding of genetics to improve bee health and other things in New Zealand. So I wiggle the thingy here. I started working with bees some years ago when I moved back to New Zealand, and we started by sequencing the honeybee genome. So by understanding all of the DNA bases in the honeybee genome, we can learn all kinds of things. And currently we're using this kind of technology to identify potential targets for novel bee-friendly insecticides on a current Ministry of Business, Innovation and Employment project. So even that little bit of basic knowledge helps us to start producing applied outcomes for industry. I'm going to talk a little bit today about our work around a gene called complementary sex determining, which is a gene which is very important in inbreeding, and we're already using this knowledge to improve breeding programs in New Zealand, so to try and control the genetics of bees. But in the next few years, we want to move ahead from these sort of small forays into using genetics to improve the industry, to start looking at techniques like genotyping by sequencing, which allows us to rapidly and cheaply understand the genetics of whole populations of bees, and then to take that information into breeding models and phenotyping to produce improved bee stocks for industry. And Gerda, my PhD student, who I've already introduced you to, so I don't need to do it again, is going to talk a little bit about that and the work that she's doing to progressing not only the technology to support whole genome selection in New Zealand bees, but also the models and the way that we deal with that data. The first thing I want to talk about is complementary sex determiner. So one of the things that we've been doing with funding from the Ministry of Primary Industries, thanks guys, is trying to look at whether we have a problem with inbreeding in New Zealand. So one of the issues with keeping bees is brood viability. So brood viability is an important aspect. One of the things that Gerda is currently doing is modelling how much impact brood viability has on honey production. But obviously, if you've got problems with brood viability, then that's going to affect the number of healthy bees in your hive and their ability to collect honey. Are you blind up here? It's really... So it's just me? Yep. Oh, okay. All right, I'm obviously adapted for Dunedin winter conditions where it's actually dark all the time. So... I'm glad that joke worked. There's a whole load of Dunedinites going. Okay, so brood viability, interestingly, depends on genetic variation in bee populations. As genetic variation decreases, brood viability in the population does as well. And the reason for this is that bees determine their sex in a very ridiculous way. Now, all of us in this room, I hope, are diploid organisms. So that means that you have two copies in each of your cells, two copies of your genome. Okay? So that's great, and it sort of means that if you've got one copy defective, you've got another copy which might be okay. Bees are almost diploid in that female bees are diploid, but male bees, drones, are haploid, which means that they only have one copy of their genome. Okay? So in all animals, animals make decisions about what sex they're going to be early in their development. So when they've just laid egg, they start reading signals, sometimes environmental ones and sometimes genetic ones, which lead to them either developing as a male or a female. And in honeybees, the signals are how many copies of the genome do they have, okay? So if they have two copies of their genome, that's fine, they're going to develop as a female. And if they have one copy of their genome, they're going to develop as a male. The problem is that actually they use variation in one single gene in the genome to determine if they've got one or two copies of their genome, okay? So this one gene is called complementary sex determiner, and if they have two alleles which are different, so two copies of that gene which are different, they're going to develop as a female, okay? Because that means they've got two copies of their genome. But if they have one copy, one allele, then they think that they're going to be haploid. And they could have one because they're haploid, or it could be that they have two copies of that gene that are the same. And as we inbreed bees, or we have smaller populations, what happens is that we get less and less genetic variation, and so there's a good possibility, or an increased possibility, that you'll end up with the same allele, the same copy of that gene in one individual, leading to diploid drones. Okay? And of course, worker bees kill diploid drones in an orgy of nastiness. So I think I'll call it that next time as well. So amazingly, in genetic terms, bees can actually count to 2. So they count the number of alleles of this complementary sex determiner gene, and if they get it wrong, if they have 2 copies of their genome, but only one allele, one variant of that gene, then they end up as diploid drones, and that's problematic. This is particularly problematic in places like New Zealand because we have had limited bee imports. All right? We have relatively small populations. With the varroa mite, we've probably lost most of our feral bees, and we have small numbers of bee breeders who are controlling their genetics. All of these factors serve to limit the size of New Zealand's bee population and to mean that we are likely to have less alleles of this gene, CSD, than we might expect. You all right there?
I'm great.
Do you want to chat? Good, fine. I like people to be comfortable, right? So one of the things we want to ask ourselves is, do we have an inbreeding problem? Because it's all very well for us to develop all kinds of fantastic tools for helping us breed bees, but if actually at the fundamental level we just don't have much genetic variation, then we have a big problem. So one of the things we did was we developed a genetic test for this individual gene, and we asked for people around the country, beekeepers around the country, to send us drone samples, okay? So drones are really easy to work with because they have one copy of their genome. So if we sequence that gene from drones, we know we've only got one copy of it, and we can learn quite a lot about it. And when we do this, we find all kinds of interesting things. So from a whole bunch of places, a whole bunch of lovely beekeepers like yourselves gave us samples. We screened vast numbers of drones. So Otto in my lab pulled the heads off lots and lots of bees and squished them. And then we sequenced the CSD gene that we found in each of those bees, and we found a whole load of different alleles. Okay? So in total, across the country using this technique, we found 72 different alleles of the CSD gene, which is a lot. It is, in fact, 60% of the alleles that are predicted to occur in the world. Okay? So that suggests that actually we don't have much of a problem with CSD in this country. But this also raised an interesting scientific question. We've got a small population, we've got limited imports of bees, why have we got so many alleles? So we decided to think again about how we might take this project further. I'm going to press the wrong button here, aren't we? So we thought again. We thought to ourselves, if we use drone bees, we're missing a whole load of diversity. So in each drone bee, it has one allele. And that allele comes from the queen, right? So every drone in a hive gets one allele from its queen. Okay? So we thought, that's a bit limiting. Why don't we do this with workers? Because in workers, there are 2 alleles. One of those alleles comes from the queen, and one comes from stored drone semen within the queen. Okay? And queens mate with multiple males, so if we were to take from a hive a whole bunch of worker bees and crush them up and then look for this particular gene, we would get a better idea of how much diversity there was, not only in the queen itself, but in the sperm that that queen was carrying. Okay? So it seemed like a much more sensible way of us to do this experiment. Unfortunately, it's also really difficult. So we had to turn to a technology called high-throughput sequencing. So when we talked about sequencing individual drone bees, we used a fairly old sequencing technique. We have to get that one piece of DNA. We can sequence that little bit of DNA, and that's fine, and we're away. Using high-throughput sequencing, we can literally sequence hundreds of millions of samples all at the same time, and at this time, these days, it's almost the same cost as doing an individual one. So by squishing vast quantities of worker bees and looking at their alleles, we decided, we thought we would find, oh, there's a diagram of how it works. It's very complicated, but it means we can sequence lots and lots of DNA all at once. Okay, this is a hideous diagram, and in fact, one of my research fellow said to me that I should never put such a diagram in front of a public audience. But you guys are smart, right, because you're beekeepers. So I trust you that you're going to help me through this thing. So this is a very complicated diagram, but all I want you to look at here, this is a kind of matrix. Up here are samples from various apiaries around the country. down here, actually the label down here, are sex alleles that we've found, alleles of different variants of the CSD gene. And in this middle here, we just have a little blue line or a little blue box which shows you how many times we've met that particular allele, okay, that met that variant. And what you can see is actually we have, and the darker blue it is, the more times we've met it. So when we look at New Zealand bee populations, there's a big chunk of New Zealand bee populations which all have the same set of variants of the CSD gene, but actually there's a massive amount of diversity in the rest of the country. Okay? So what we discovered was a huge amount of diversity in the CSD gene, and in fact we've now identified over 180 alleles, which is 90 more than have actually been predicted to occur in the entire world. So those predictions were done by Germans who were clearly wrong. I didn't say all Germans are wrong, just these particular ones. Sorry, by Europeans who are clearly wrong. And it's an indication that actually the rate that these alleles evolve is kind of much faster than we think it might be. Okay, so this is cool. New Zealand has no lack of genetic diversity across the nation. We have all the bees we need and all the genetic diversity we need to do interesting things. We can take this information and we can display it in all kinds of ways. I want to show you something even more complicated, and that's a family tree of those alleles of those genes. Okay. And the thing I want to point out to you is we can take all those sequences and we can look at how closely they're related to each other. And when we do this, we find a really remarkable thing. Outlined in red here is a whole group, a whole family of this gene, which comes only from New Zealand. We're comparing New Zealand sequences with sequences from around the world, and New Zealand has its own special ones. In the last sort of 200 years that we've had bees in this country, it seems that we might have evolved our own alleles, and that in fact, the CSD gene is one of the fastest evolving genes that we know about in animals, which is great news for us, right? It means that while we need to worry about inbreeding our bees, particularly when we're looking at closed populations or breeding populations, actually in New Zealand there seems to be a vast diversity of alleles out there, and that those alleles seem to be changing all the time, we think. So, I've got implications. There's implications. We have lots of CSD allele diversity. We do have to worry about closed breeding programs. So if you're doing artificial insemination technologies, then every generation you breed through, there's a good likelihood you will lose some genetic variation at this particular gene. You need to come and talk to us because we have tools which will help you assay how those CSD alleles and help you control them. We have information which suggests that actually, unlike as the Germans think, CSD evolves very fast, which is good news, and that we have lots and lots of diversity in bees in New Zealand, right? We might think that the situation in New Zealand is that we have very small genetic diversity, very narrow populations, but in fact our studies suggest that actually bees in this country have come from lots of different places, there's lots of genetic diversity, and that provides an opportunity, okay? The diversity that we have there underlies traits, some of which are important for production. And it gives us the opportunity, if we have lots of genetic variation, to use artificial selection to select lines of bees to do particular jobs. So there is variation in honey production, disease tolerance, and pollination traits, and some of that variation is to do with genetics. And we have a current Ministry of Business, Innovation and Employment bid to develop a pipeline available to the whole industry to use genetic techniques, genomics, to improve bee structures. Now, we haven't got the funding for that yet, so if you're sitting on a plane next to somebody, go and tell them how important bee breeding is, because it may be that they're going to make the decision. But while we're waiting for that decision, Goethe has been working with a Callahan-funded PhD program, funded by a bunch of industry partners, to start develop some of the tools and some of the ideas that we're going to need to talk about genetic improvement in bees. So I'm going to pass over to her. Have I given you enough time? Yeah, whatever. Go.
Yeah, hi. I'm probably not going to be as funny, but I'm German. I guess we're not supposed to. So.
That was the biggest laugh.
I am going to talk about genetic improvement in general, and I'm only going to touch on the work that I am doing with the Taylor Pass Honey Company and Abacus Bio, because I believe that we need to actually explain what we're trying to do before we're trying to do it. So genetics, what exactly are we looking at? We always have to keep in mind that the phenotype, what we're seeing when we're looking at an animal or at a plant, is only a result of a combination of the genetics and the environment that the individual happens to be in. So all that I'm going to talk about is this G in the middle. I'm not talking about how things are going to get changed by management decisions. And it's just something that I need you guys to keep in the back of your mind. So if we're looking at genetic improvement, we always have three core components. The first one is the value proposition. So everything that we're selecting on, every trait, every characteristic that we choose to select on needs to actually be relevant within the system and within the market that's targeted. So we also need to keep in mind that it needs to be simple to implement. So if I'm asking Taylor Pass to keep records on something, I can't just make stuff up and say, oh yeah, just come up with this really complicated measurement for something. I actually need to say, well, this is what I want to know. Can you please tell me how much honey is produced per hive? So the third part is then the actual design of the breeding program, which once again needs to be tailored both to the operation and the industry and to the market that's targeted. So the value proposition. I have an example here. This number, 300 million per year of genetic gain, once again, I'm just talking about the G, I'm not talking about the environment. That's the New Zealand dairy cow industry. And that's what's happening right now. Right now, in the New Zealand dairy cow industry, each year, 300 million are gained in performance potential by the animals that are in the national herd. So how does this happen? Well, basically, there are a couple of things about genetic improvement that are quite important. First of all, it's permanent. If you improve your stock, if you improve the genetics, and with the genetics the potential for performance, that's something that you're going to keep. Unless you go ahead and actually squash all of your improved queens at the end of the season, and potentially even then, because they've already disseminated some of their genetics, that's not going to go away. So it's also cumulative. Every year, if we keep improving genetics, we are going to actually also improve the performance potential of the population that we're looking at, and it's cumulative for multiple benefits. So all of these steps, they're not just, we're still in the dairy industry here, they're not just improvements in milk yield every year, there might also be structural traits, survival traits, calving knees, just general stuff. So we can kind of tack on several different traits, several different layers of genetic improvement over time, and it's all going to add up. So what happened in the dairy industry is that these are the trends for the different breeds that are being used, and even though they kind of differ a bit in terms of where they are, this is breeding worth. This is the actual dollar value of their genetic material. So you can see that even though they're quite different in their level, they all have the same trend. So they're gaining about $10 per cow per year. And if you add in that they've been doing this is a 10-year analysis. So if you add in that they've been doing this for 10 years, they've been adding improved heifers every year. They've been adding about 750,000 heifers a year. You end up with that number of 300 million a year that are being added right now. So the next step. Implementation. In order to actually put something in place, we need to figure out what do we have to begin with. So we need to record a lot of stuff. We need to record parentage, and we need to record the traits that we're going to modify. So in order to modify the genetics, we need to know where they come from, and we need to know how are the genetics that we have right now actually performing. So if we're looking at, for example, honey production system, and this is quite simplified, just because I'm trying to make a point here, we need to ask a couple of different, very important questions. Where does profit come from? Where do costs come from? What's the particular task that we're asking the animal to perform? And what would the ideal animal, if we're taking all of this into consideration, look like? So the answers to these questions are, well, high honey yields. Cost is mainly labor. And if we consider that, then we want an animal that is supposed to produce honey in high yields and is also gentle. So we're reducing labor costs by reducing time that we have to spend with them. So in this system, we have to record three things. We have to record the queen line, because we can't really record the drones. We have to record honey yield and gentleness. So if we're talking about how we're going to do this, there's another important question, and that's how fast and how cost effectively can we actually get to where we want to be. So we've got a couple of tools that have been used quite successfully in other industries, and we are in a very, very comfortable position because we can look back and say, okay, this has been done in dairy, this has been done in beef, this has been done in sheep, or even in ryegrass or pines, and we can use these tools with the limitations that we know they have because other people have failed. So some of these tools are progeny testing, so not only just looking at one individual, but looking at their offspring and actually feeding all of the information that we can get back into the system just to get that much better quality of data on one specific queen in this case. One thing that we could potentially do that has been done quite extensively in sheep and in the New Zealand deer industry, for example, is crossbreeding. In bees, crossbreeding is a bit tricky because a lot of hybrids between subspecies, for example, Africanized bees or Russian hybrids, actually turn out quite aggressive. So we might be able to just implement line breeding instead, just establish lines within a subspecies, just cross different lines of Italians, for example. And the last thing that is right now kind of where we want to go, we can actually use DNA-based technologies to better navigate what we're trying to do with the progeny testing, with the line breeding, with the entire system. So what are DNA-based technologies? Basically, we're doing what Peter was talking about. We're looking at the DNA, but we're not just looking at this one specific gene, we're looking at the entire genome. And we can use this to perform genomic selection. Genomic selection basically just means that instead of looking at the animal and saying, this is the animal, this is what it looks like, this is how much it is worth to the population, we're actually looking at this is the animal's genome, this is its DNA, and this is what it's worth to the population. And then we add that into a system where we already know what the phenotype, what we're seeing, is worth. And we can utilize both of those so much better just for being able to combine them. We can also track desirable genotypes, which means that we know this is a really amazing queen. She has outperformed everyone else over the last 3 years. Then we know this is her genome, this is what her DNA looks like, and we can then compare that to other queens that maybe we don't really know that much about because they just hatched. And we can say, okay, we can actually see this reflected in this other younger individual. This virgin probably has the potential to outperform everyone else. Let's just use her instead of another animal. And we can actually establish full parentage. So now we don't have to necessarily use AI to know which drone we're looking at as a father. We actually look at the DNA and then we can just say, well, this is what we have, and this is how related it is to everyone else. So something else that people keep talking about or keep thinking about when someone mentions genomic selection is marker-assisted selection. Marker-assisted selection means that we define a trait, we define a characteristic, and then we look at the genome of all the animals that have this, and we say, well, this is something specific within that genome that all of them share. This must be a marker for this trait. Unfortunately, with bees, we don't really know enough about what the entire population looks like to say, we have these markers, let's just use them. So we have to be very mindful of that, and we have to be careful if we're using markers that we actually still look at the animal itself. And then the last bit, and that's where Peter's talk comes in, is the control of relationships. We need to control inbreeding, even though we have a pretty diverse population in New Zealand. We don't really have the same diversity necessarily in every operation. So we still need to just look at the structure of the population in question and actually manage what we see there in order to maintain diversity. Also, genomic relationships can actually help establishing lines within a population. So being able to just capitalize on hybrid vigor, on crossbreeding itself, would be quite good. So just to kind of wrap this up, this is a graph for pines. So that's where the volume comes from. But nevertheless, this is a basic principle. So the normal genetic improvement that was performed in New Zealand pines is the black line. And at some point, they implemented genomic selection, and they achieved this massive lift. In bees, I would expect this lift actually to be even more significant just because the inbreeding control and the control of our relationships is so much more important. That's it.
Next one.
So.
Okay.
Thanks very much. So we need to thank a whole bunch of people. Thank you, Gerda, that was awesome. Good job. I think she did well. I think she deserves a clap for that. You obviously don't. Otto, who I don't know if he's here today, but Otto did all of the work on CSD, so I was describing his work. Better Bees Research, who have helped us with bees for a long time and who have been awesome in helping us implement some of our weird ideas around bee breeding. Peter Fennessy and the Taylor Pass Honey Company and Abacus Bio, who are partners in the project that Gerda was talking about. All those people who gave us samples, and the Ministry of Primary Industries through the Sustainable Farming Fund for support, and thank you all for listening to us. We've got time for some questions, and I'll be really disappointed if I don't get any. I may even cry. Sorry, you can always rely on him.
Absolutely. I'm going to ask you a patsy question, Peter, because as a researcher, you can never get enough of what I'm going to ask about. But what I'm going to ask you about is our industry's commitment to the funding of this kind of work and what we should be really seriously looking at in the future when we use models like dairy and sheep and other industries.
So that's a really difficult question for a scientist. There's two ways to answer that. The first one is to say you should be funding research, and that's great. And I'd get more money, and that'd be wonderful. And so would my colleagues, because there are awesome scientists in New Zealand. To tell the truth, we do have an awesome science system of people interested in bees in this country. It's diverse. We've got really interesting people working on lots of different projects, all of which I think are focused quite clearly on good outcomes for the industry. I know it's very hard to see sometimes when you're a small operation who's close to breaking even that actually there is a value in doing research, but there is, and that value has been shown very clearly in the dairy and sheep industries, where genetic gains in particular have made enormous differences in their industry. I think that bees are a really interesting system for geneticists. Firstly, they have interesting genetics, right? And so a lot of what we do is about their basic biology, and I would have loved to have told you about some of that. Maybe you can come back and I'll talk about that next time. But it also gives us an opportunity, because the difficulty of their genetics has meant that selective breeding in bees has been slow and it hasn't been taken up very much. And that gives us huge opportunities. It means actually we are at the start of the possibility of genetic improvement. And in all of those graphs that Gerda was putting up, the biggest gains happen early on in these programs. So I think that there's a huge value to doing research. What we do have also is a supportive government sector who are very keen to help support bee research and to put funding into it. And we've been able to get funding for those kinds of projects. But more and more we're asked to find industry co-funding, and that is quite difficult. So if you would like to, I think that there is value in doing this kind of research, and we are very clearly focused on providing outcomes for your industry. So maybe it's time to think about whether there's a way that the bee industry can help co-fund some of this research in partnership with government. Yeah, go on. Fair enough.
Something that we also have to kind of keep in mind is the fact that part of the reason that the other livestock industry in New Zealand are performing very well, we're performing very well before dairy kind of crashed, is that they have centralized systems to manage their genetics. And that doesn't mean that everyone is in the same breeding program. It just means that there's a central system to manage the information and actually capitalize on all of the little bits of information that are collected somewhere on farm. And in my mind, that's something that should be possible for bees as well. We also need to keep in mind, however, that this is still an emerging, from my point of view as a livestock geneticist, this is an emerging industry. This is an industry that has gone through massive changes over the last few years. And in emerging industries, livestock industries, there's always going to be just a couple of operators that push things. So watching other operators that actually invest in genetics closely might still be a valid step for people that are not really committed to fund something themselves, and see if they can kind of hitch onto that, because that's what's going to push it.
Hello.
Thanks. Yeah, love the work. I think it's very interesting. Just one question, I think maybe related to, for example, if you are trying to improve sheep or cattle, there is more control who they're mating with, but once, if you select bees, even for example for a varroa tolerance, once you release those queens, then they start mating with, and you don't have to control. So what is your perspective there?
I mean, this is a key problem for us, and there's kind of a bunch of different answers to this question. The first is that actually maybe if we all engage in selective breeding, then the quality of bees and the quality of drones that are out there will improve. That's got to be the long-term aim, right? That actually your queen is going to mate with a drone carrying Varroa tolerance-related genes because that's what's present in New Zealand. And I think that when you look at the sheep industry, that's exactly how they've driven this process, that by driving the breeding value of sheep to individual sheep breeders, you improve the national flock without nationally controlling the whole process. So that, I think, is the long-term endpoint. And more important, before we need to do that, we need to think about artificial insemination and its value and the way that it's used to ensure that we're at least controlling in breeding programs the quality of the bees, controlling both sides of the genetics. But once those go out into operations and form mated queens, then obviously we have a difficulty with that genetics. We're also in this program really interested in looking at the flow of those genetics. What happens to the genetics of a breeding program when it goes out into a large operation? How does that affect individual hives? And so we probably will be able to see gains even if we're not controlling the drone cloud in the future. But we need to quantify that so that when we're talking to beekeepers, we can show them exactly what the value of using a breeding program stock might be. So it's a really good question, but it's something that's one of the unusual things about bee genetics that we have to deal with, and we're struggling with that at the moment, or at least she is. Yeah. Are we done? We're done. Sorry.